Good morning, students. Today we are going to talk about experimental evidences to prove DNA replication. So again, a very important topic of this chapter, molecular basis of inheritance. But before continue with this chapter, let's take a recap what we have discussed yesterday. Children, yesterday we have discussed about the different mode of replication which are suggested by the scientists. That is conservative mode, dispersive mode and semi-conservative manner. So actually DNA replicate, there are three theories which were proposed by the scientist time to time. Like what happened in the conservative mode of DNA replication, like entire DNA act as the template, and from entire DNA a new DNA is synthesized. Like here the red color DNA is the parental DNA, from which a new DNA, blue color DNA, is being synthesized. In the dispersive manner, what is the dispersive mode of DNA replication? In that mode of DNA replication, not the entire DNA act as the template, nor the single strand act as the template. The parental DNA is alternate with the newly synthesized DNA. Like one portion, red color is the parental, then blue is the newly synthesized. Again red is the parental, again blue is the newly synthesized. So this way of DNA replication is known as dispersive DNA replication. What happens in semi-conservative DNA replication? In semi-conservative way of DNA replication, in the DNA, because DNA is the double-stranded structure, a single strand acts as the template, the parental strand. And from that parental strand, because there is the complementary base sequences, so the second strand is being synthesized, right? So here red color strand is the parental strand and blue is the newly synthesized. And this semi-conservative is the most acceptable theory, and it is accepted or proved by the other scientists. So then now we are going to talk about some of the experimental proofs which actually prove that DNA replicate in the semi-conservative pattern. As we have discussed about all these patterns, then came an experimental evidence to prove how exactly DNA replicate. So this experiment was performed by Meselson and Scholl, and that is why it is known as Meselson-Scholl experiment. So what was done in this experiment? Let us go to understand this experiment. So this experiment was to prove the same semi-conservative nature of DNA replication. So by now we already know what is semi-conservative nature. So what was done in order to prove that? We will see. Now children, it was experimented with the bacteria E. coli in 1958. So here on your screen is the diagram of Escherichia coli, named as E. coli. Now what was being done was that, first of all, even before experiment was carried out, E. coli were grown in two different medium. So one set of E. coli was grown in medium with N15. Now nitrogen has two isotopes, N15 and N14. So atomic mass is different in these cases. This N14 has 14 and N15 has 15 atomic mass. However, none of the isotope was radioactive. Both of them are non-radioactive. Now children, first of all, in the very first step of this experiment, E. coli was grown in medium containing heavy isotope, that is N15. Do you know heavy isotope? Okay, I will tell you. Children, in nature, some elements have different isotopes, which they differ in the number of neutrons. The difference in number of neutrons make the isotope atoms heavier, right? Now children, in this experiment we have two isotope of nitrogen, N14 and N15. And if you compare these two, N14 is the one which is a more abundant isotope, where there are 7 proton and 7 neutron, and it weighs around 14 gram per mole. Talking about the N15, N15 is a little less available compound than that of N14, and it contains 7 proton and 8 neutron, and it weighs around 15 gram per mole, right? The question is, why heavy nitrogen isotope N15? As I just mentioned, that the very first step of this experiment is that E. coli was grown in medium containing heavy isotope N15. N15 में ही पहले क्यों किया गया? Why heavy nitrogen isotope N15 is used first in this experiment? Remember? Yes. Because children, DNA bases contain nitrogen, right? Also another distinction is that yes, we know nitrogen is the major constituent of DNA, right? So if nitrogen is being replaced, so the DNA will have changes. अगर नाइट्रोजन में कोई भी चेंज होगा तो डीएनए में भी चेंज होगा, क्योंकि डीएनए का इंपॉर्टेंट पार्ट होता है नाइट्रोजन, क्योंकि डीएनए में नाइट्रोजन बेस इन्वॉल्व होते हैं. फॉर एग्जाम्पल, इफ द नाइट्रोजन इज रेडियोएक्टिव, then the DNA will become radioactive. Now if the nitrogen is non-radioactive, then the DNA will be non-radioactive. तो नाइट्रोजन में अगर कुछ भी चेंज होगा तो डीएनए में भी वो चेंज तय है, निश्चित है. क्यों? क्योंकि डीएनए का इंपॉर्टेंट पार्ट होता है नाइट्रोजन, इंपॉर्टेंट कंपोनेंट है नाइट्रोजन, क्योंकि डीएनए नाइट्रोजन बेस से मिलकर बनता है. If nitrogen has N15 isotope, because nitrogen is the part of DNA, so it has been observed that DNA with N15 isotope is heavier than that of N14 isotope. Now what was done in this case, where the set of E. coli grown in the medium with N15, right? क्या होगा? पहले हमने क्या कहा कि E. coli को N15 मीडियम में ग्रो किया, तो क्या होगा इस केस में? So it is very obvious that E. coli which are grown in this medium, the DNA of this set of E. coli will have N15, जो आपको ब्लू कलर से दिखाया. नाइट्रोजन 15 में जो E. coli बैक्टीरिया ग्रो किए गए, उन सभी बैक्टीरिया के DNA कैसे हो जाएंगे? N15 वाले. As a result, this DNA will be heavier than that of which are grown in the medium N14. तो इसके DNA भी जो है वो heavier हो जाएगा. So there was another set of E. coli which was grown in a medium of N14. So here again the DNA of this E. coli with N14 जो green color से दिखाया है. So it was observed that N15 DNA is heavier than N14 DNA. Now children, how to get it that N15 was heavier and N14 was lighter? How do we know that? So for that we have to perform the process of centrifugation. Okay, so what happens in centrifugation? Centrifugation में क्या होता है? If a mixture is rotated at a very high speed, what happens? Heavier particles tend to settle down just close to the center. जो heavy particles होते हैं, they are settled down. जो इसमें sediment आपको दिखाया गया है, और ये जो diagrammatic representation है, इसमें जो blue color का जो center part है, that contains N15 because it is heavier. The lighter one moves farther than the center, और जो lighter one होंगे because they have less density, they moves farther from the heavy densities, means at the surface. तो इस डायग्राम में अगर आप देखते हैं तो वो सुपरनेटेंट से आपको दिखाया गया है, राइट? और अगर इस डायग्राम में देखते हैं तो N14 जो है वो ग्रीन रीजन, राइट? So this green color denotes N14 and this blue color denotes N15. So here the circumference you see denotes the presence of N14 DNA and this blue color denotes N15 DNA. So N15 was closer because it was heavy and N14 was farther because it was lighter. इसीलिए N15 was found to be heavier than that of N14 DNA. This is what was observed with centrifugation. So basically हमने क्या देखा? If the isotope of nitrogen is different in two DNA, then we can distinguish DNA by their weight. If it's heavier, that means it has N15. If it is lighter, that means it has N14. So this is going to be the basis of the experiment, which is performed by Meselson and Shaw. Okay? तो बेसिक्स जो था वो मैंने आपको बता दिया कि मेजल्सन एंड शॉल ने जो एक्सपेरिमेंट कंडक्ट किया उसकी बेसिक थ्योरी क्या थी वो मैंने आपको बता दी. सो नाउ लेट अस लुक एट द एक्चुअल एक्सपेरिमेंट. व्हाट दे एक्चुअली डिड. नाउ लेट अस लुक एट द एक्चुअल एक्सपेरिमेंट. द वेरी फर्स्ट स्टेप. Grow bacteria in a media containing the heavy N15 for many generations. As a result, all the bacteria has N15 in their DNA. The second step is transfer some N15 bacteria to grow in N14 media and allow to grow for several generations. The result is DNA of newly divided bacteria will have N14 instead of N15. Third step, the sample taken from the growing bacteria at different time and study the density of DNA of the cell. Right? So what happens actually? Simply the idea to mark the old DNA and see what happened to the newly synthesized DNA. Now children, let me explain you what happens in the actual experiment. So they allow E. coli to be grown in medium N15 for several generations. So when we say several generation, it is like you start from parental generation, then you have F1 generation, then F2 generation, and the process continue for the several generation with N15 medium. So what happened? All the E. coli which are beginned produced in this medium will have DNA with N15. तो आपने इस डायग्राम में देखो रेड कलर से दिखा हुआ है. तो सेवरल जनरेशन तक जब E. coli को N15 मीडियम में ग्रो किया गया तो हर बार E. coli का जो DNA है वो N15 था. It has not about blue and green or red or blue, red or green. Only the color I used to explain, right? पहले मैंने blue और green लिया था, अब red और green ले लिया. So the color is only to explain you. Again, E. coli with only N15 in their DNA were transferred to the N14 medium. So now these E. coli has green color. अब इनमें जो DNA थे वो कौन से कलर के थे? ग्रीन कलर के, N14 वाले. Now these E. coli with green color, now these E. coli with N15 with red color are transferred to the medium has N14 with green color. So now when they are grown in N14 medium, will they have green color? Will they have red color? They have green color because they grow in the N14 medium for the several generation, and now the DNA of those E. coli become green. तो N15 वालों का red और N14 वालों का green. How will it be? Now E. coli cell will have allowed to divide even for multiple generation, but the sample of E. coli were taken periodically in between. Like after one generation it was taken, again after generation 2 E. coli sample were taken just to see what are the changes taken in their DNA with each generation. हर generation के बाद उनकी DNA study की गई और देखा गया कि क्या changes थे. So after generation 1, how is the DNA? After generation 2, how is the DNA again? After generation 3, अब DNA में क्या change है? Like this. For this, you collect sample periodically. After some time, there is the sample collection. Need to collect the sample. Like if you allow to grow E. coli, you need to collect the same sample after some time for the study. Now it was seen that for complete replication, E. coli takes 20 minutes. तो यहाँ पे आप देखो कि बीच में 20 minutes का gap दिया है. Because E. coli takes time to grow around 20 minutes. I mean, first sample you have to take after 20 minutes, right? Next generation produced after 40 minutes and so on. Similarly, you can collect samples, right? And samples were taken and DNA was extracted periodically as cell division continued. So this cell division kept on continue over a period of time, but it ensured the sample is being collected for every generation. So while you sample collected, you get to know what kind of DNA are being generated in each generation, right? So this was the experiment actually performed. So now the question was, what kind of DNA produced in each generation? So let us see what were the result. So children, in generation 1, when I say generation 1, that means after one complete round of replication. That means after 20 minutes. So what was seen in that sample, that how do we know how is that DNA? Yes, exactly, by the centrifugation जो हमने बात करी थी. DNA की जानकारी कैसे मिलेगी? Centrifugation से. Because now we know if the process of centrifugation is being performed by looking at where the DNA sample is dispersed. We can actually see whether it is N15 DNA or it is N14 DNA. For generation 1, it was found like DNA was found to have intermediate density after first replication. So it is clearly seen here that the density is neither matched with N15 or nor with the N14. So it was a in between N14 and N15. That means it has mixed of both green and red, right? So that is conclusion after first generation. So that means generation 1 has both the colors, red and green, N14 and N15 both. So that is why their density will also intermediate between two values. What happened after in the generation 2? The sample collected after 40 minutes. So here it was observed that equal amount of DNA with two different densities were found. So here density was not intermediate. Half of the sample with red color and half of the sample with green color, right? So what like this? This is the structure. This is the result we found, right? Half of the sample have density with the green color and remaining half of the is with intermediate red and green both, and also with the green color, right? अब इन चारों में से आप देखोगे तो दो जो है वो ग्रीन कलर के हैं और दो जो है वो इंटरमीडिएट है. That is why I said that half are the intermediate and half for the green color, right? That means N15 density is all gone. कहीं रेड कलर नहीं ज्यादा दिख रहा है. What is left over? Intermediate density 50% and N14 density 50%. So this was the observation. Now children, let us look in the detail at the experiment and the expected result of each hypothesis. Right? So this is the experiment which was done by Meselson and Scholl, that N15 and N14 is taken, and after the interval of every 20 minutes the samples are taken and the study of DNA is carried out by the help of centrifugation and take their observations and results. Now simply observe all those hypotheses and results. Acha, the first thing is that can you relate all these observations to the experiment which was performed by Mendel with the pea plant? अभी जो हमने बात करी है सारी के सारी मेजल्सन एंड शॉल के एक्सपेरिमेंट में N15 और N14 की, तो आपने देखा अगर हम इसे कंपेयर करते मेंडल पी प्लांट के एक्सपेरिमेंट से, there also we saw similar sort of output in F1 generation and F2 generation, right? सेम ही तो दिख रहा है. तभी क्राइटेरिया में हमने डिस्कस किया था कि जेनेटिक मटेरियल शुड बी फॉलो द मेंडेलियन कैरेक्टर. रिमेंबर, जेनेटिक मटेरियल के कैरेक्टर हम लोगों ने डिस्कस किए थे. अब ये जेनेटिक मटेरियल भी तो जो है मेंडेलियन कैरेक्टर फॉलो कर रहा है. दैट्स व्हाई व्हाट इंटरेस्टिंग थिंग डिड यू ऑब्जर्व हियर? Children, here we saw that conclusion after generation 1 and generation 2. So let us look at the conclusion in little more detail. So in overall experiment we started with the red color DNA, then we found in the F1 generation that all the individuals were intermediate with N14 and N15, and in F2 generation 50% carries N14 and 50% carries intermediate. So quite surprising results. But what this result tells us about the type of replication or the mode of replication of DNA. Children, the first thing that was observed here, that intermediate density existence, hybrid, mixture of both, right? And presence of hybrid told that it was not conservative hypothesis. जब हमें intermediate मिल गए, तो इंटरमीडिएट मिलने का मतलब सीधा सीधा ये हो गया कि कंजर्वेटिव मैनर में तो रेप्लिकेशन नहीं होता, क्योंकि कंजर्वेटिव हाइपोथेसिस के अकॉर्डिंग इधर कंप्लीट रेड होगा या कंप्लीट ग्रीन होना चाहिए, हाइब्रिड नहीं होगा. सो वन थिंग इज क्लियर, ओके, दैट मोड ऑफ रेप्लिकेशन इज नॉट द कंजर्वेटिव रेप्लिकेशन, राइट? क्योंकि कंजर्वेटिव का रिजल्ट ये होगा. In presence of N14 DNA in generation 2, it shows that it cannot be dispersive as well. अब F2 generation में हमें N14 मिल गए, right? इसका मतलब ये dispersive भी नहीं है. क्यों? Because each DNA should be mixture of both. पर ये complete green भी है, तो complete green मिल गया, इसका मतलब dispersive भी नहीं हो गया. अगर हम dispersive से देखें तो हमारा रिजल्ट कुछ ऐसा आना चाहिए. अब बचा क्या? बचा सेमी कंजर्वेटिव हाइपोथेसिस. सो इट वाज फाउंड दैट सेमी कंजर्वेटिव हाइपोथेसिस कुड एक्सप्लेन द एंटायर एक्सपेरिमेंटल रिजल्ट, राइट? तो सेमी कंजर्वेटिव के अकॉर्डिंग रिजल्ट्स ये आए. अब अगर याद हो तो हमने जो डिस्कस किया था वो ये डिस्कस किया था रिजल्ट, राइट? सो इट इज क्वाइट सिमिलर विद सेमी कंजर्वेटिव मैनर. So it suggests, by this experiment, it is suggested that DNA replication is semi-conservative. So finally it is seen. तो अभी तक हमें पता है कि DNA replication is semi-conservative nature. But now exactly replication takes place. क्या क्या रॉ मटेरियल रिक्वायर होते हैं, रेप्लिकेशन कैसे होता है, ये सब देखना है. इफ यू वांट टू प्रिपेयर अ केक, यू नीड रॉ मटेरियल फॉर इट. यू नीड फ्लोर, as essence, butter, water, everything you need to prepare the cake. So similarly, in order to understand DNA replication, we need the essential things. तो वो सारी essential things हमने अब डिस्कस कर दी. अब हम नेक्स्ट वीडियो में डिस्कस करेंगे chiefly DNA replication. So in the next video, we will meet again with an important topic, with an important discussion on the topic DNA replication. So the important thing for you is to revise and learn the today's topic. So children, till then, have a nice day and thank you.